they are able to survive or actually are a dead end. The majority of these changes are actually a dead end, but some causes a benefit to the virus. So that process of changing and selection and so on, we call that viral evolution.
You say a dead end or beneficial. What are those two outcomes for a virus? What does that mean for in the virus world?
Yeah. So that's a good question. If you think of it, the virus is really trying to find a balance. Because if it makes too many changes, then it's unable to reproduce itself. It just doesn't work anymore, so to say. But if it just makes enough changes, then it can adapt to its environment. So it's really finding that right balance for the virus.
Now, there's no brain in a virus. The virus isn't thinking itself through,
Right.
so it's just a process where it replicates, and then it's shifting as it replicates in a way.
Yeah, that's right. No, the virus doesn't have a brain or eyes or ears, but still it reproduces a lot of itself. And by doing that, it makes a lot of errors. Having said so, there's also a mechanism that this virus has is called proofreading mechanism. So every time it makes a copy of itself and it makes an error, or you can think of a typo in a sentence or in a word, this error is then corrected and replaced with the correct letter. So this is a mechanism that the virus has to make sure it doesn't change too much. But it does make a few changes, and we're closely monitoring those changes to see what their impact is.
So you're speaking about, now are you speaking about the virus that causes COVID-19, the disease? You're saying that one has this kind of proofreading mechanism, or do all viruses have this proofreading mechanism as well?
Well, in the group of viruses that COVID-19 is part of, it's only the ones that are quite large in terms of their genome. So this is a bit technical, but the virus is 30,000 letters long. Because it's so long, the virus really needs some help to making sure that whenever it copies itself, that it does that correctly. But as I said, it still makes some errors, and those errors may have an impact on viral countermeasures or how the virus behaves or what the virus will look like.
So we'll keep talking about that, and please ask your questions for our expert here. Anything you want to know about how viruses change. Peter, I wanted to bring you in now as well. So we talked about the virus changing itself. Recently, people spend a lot of energy and attention watching mink and human interactions in Denmark because of some changes happening there. Can you give us the background story a little bit and why everyone, including WHO, was so concerned about that?
What happened is that the virus jumped from humans to mink and started to infect mink in different mink farms. And it was allowed to spread among those minks, and in that process it was allowed to have these opportunities for changing and for mutating. And we started to see some of these changes, some of these new variants of the virus jumping back to humans. And that was the big worry for us, because when we see that, we are of course afraid that one of these variants, one of these changes, one day would be a nasty one for us, and we'll start another series of problems with the new variant of the COVID-19 virus.
One thing you helped me understand a lot as I sat in on meetings with you when you were explaining this is that when the virus is in humans, we're all kind of similar in some ways, and the virus changes a certain amount when it's in humans. When it's gone into mink, and mink being so different from humans, there's sort of pressure on the virus that it needs to change more, and therefore when it comes back into humans, we might be looking at a much more different virus. Have I captured that?
Correct. Remember that the COVID-19 virus that we know very well by now is a fully human-adapted virus. It's a human virus now, and therefore it doesn't need to adapt further. It doesn't need to change when it circulates in the human population. It has a perfect environment there. But when you put it in the mink population, as you said, it's a new environment, and it tries automatically, as Frank was explaining, to adapt to these new environments by making a few changes. Of course, it's not an active process, but it's just a viral, biological process that happens naturally.
Like a evolution, like a natural selection
Natural
of one
selection.
is stronger, it will do better than another variation.
You will have all these random mutations and changes, and nature will select the ones that are most appropriate, most beneficial for the virus in this new environment.
This is a question for you, Frank. Do symptoms necessarily change when the virus changes? It's a question from Facebook. The way our friend put it is, do symptoms change when the virus mutates?
Yeah, so we've been actually following this quite closely to determine if different kind of mutations have an impact on disease progressions or symptoms. And actually, so far, what we have seen, it doesn't seem to cause any differences in symptoms. So it's just the same clinical presentation or symptoms as you would see with your quote-unquote normal SARS-CoV-2 or COVID virus.
So there's a few other similar questions. You're talking about symptoms, but people also want to know, there's a question on LinkedIn, for example, what are the risks that as it changes over time, as it passes around even amongst humans, that there'll be an effect on the vaccines, the potential vaccines that are still in development, and how effective the vaccines can be?
Right. Well, remember what I just mentioned. The virus is relatively stable, and on top of that, it also has what we call this proofreading mechanism. Nevertheless, the virus does change, and depending on where in that genetic material or in the recipe of the virus that change takes place, it may have an impact on what the virus looks like, the shape of the virus. Because remember, we're talking about this coronavirus, which is sort of like a ball with protruding pins coming out of it. And these pins, if the changes happen in that pin, it may look a little bit different. So the possibility exists that it may impact the virus, may impact the vaccine. But so far what we have seen, the virus is relatively stable. The vaccine is also developed using the existing virus. So we haven't seen any red flags just yet. But as I mentioned, we're closely monitoring this with a large group of scientists. In fact, we've been monitoring this right from the start, ever since the beginning of the outbreak, working with a large group of international scientists from all over the world. We have regular calls, discussions, working groups, and so on. There's a lot of activity in the laboratories that we see, looking into what do these changes really mean. And just recently, after we've always been looking at it, we've also started to have a bit more structured working group approach to these changes in the virus. And this is a more dedicated working group that we have established, and that is what we call the SARS-CoV-2 Virus Evolution Working Group. And with this, we can take a more systematic approach at looking what these changes mean and how we're going to tackle them and better understand them.
I think what you explained off the top as well is that the tests to detect this virus and the vaccines to combat it are designed around a specific virus. If the virus changes a lot, then you worry that the tests won't recognize that virus and the vaccines won't work against that virus. But to summarize what you've said so far, the virus doesn't seem to have changed in any important way that would have an impact on the vaccines that have been in the works so far or on the diagnostic tests. So that's good news, but we continue to monitor closely should there be any big shifts.
We need to remain vigilant.
And not just vigilant, I mean really active. It's
Yes.
like an active kind of following up. Peter, a question from Lashika, who wants to know, do we think that the virus is natural or man-made? What do we know about the virus? And you might come in on that as well, Frank.
What we know is that when we look at the makeup of the virus, is that it has all the elements of a natural virus. There is nothing to indicate that it will be man-made. And you can try to think about building a car with Lego blocks or something like that. And if it was built with known blocks, for example, with all red or blue blocks, when you look at the final product, you will recognize that it's made by these blocks. But here we have not that picture. We cannot recognize elements that would have been used to construct such a virus. So it's, everything indicates that it's a natural virus.
That having been said, we are still trying to understand how this virus came from the animal world, which is where a natural origin would be, into the human world. And Peter, you're working very closely with the international group of experts that WHO has convened to do this virus origin studies. Can you tell us a little bit more about what that process will be? How will we go about trying to understand that recent history of the virus and how all this began in the first place?
What we try to do is to figure out what happened last year before the first case were detected in December 19. You remember the first case were detected in Wuhan in central China, and after that we all know the history and the course of the pandemic. But before December 19, we don't really know what happened. We don't know how the virus jumped from its most probable natural environment in bat population over to humans. And it's that bit of the history that we need to reconstruct and understand and try to find. And the process to go about that is to, what we would like to do with the international team and counterparts in China, is to go back in the Wuhan environment, re-interview in depth the initial cases, try to find other cases that were not detected at that time, and try to see if we can push back the history of the first cases, if we can find all the cases that could have important information to tell us about where they may have picked up this virus. We should also look again at the famous wholesale markets where the first cases were detected. Look at everything that went in and out of that market at that time, and try to find out where these products and animals and food products came from, where the workers working in this market who got infected severely in December, where did they came from? Did they all have some commonalities somewhere outside Wuhan in some of the countryside or provinces or farming environments or hunting environments in southern China, or perhaps even outside China, and then concentrate in these locations, concentrate in these environments, and try to then there conduct more in-depth studies, survey a number of animals, a number of humans in that area, and try to see if we can find some clues that would tell us the early history of this virus.
So it's a kind of an epidemiological work, and very human as well. You talked a lot about needing to talk to people and understanding kind of the background of their work and their life, their exposures, and so on.
Yes, and it can be both time-consuming and quite complicated, because it's about trying to recall the memory of people in that period, and it takes time, it takes special techniques to get that kind of information out of people, and then connect the dots. You might get bits and pieces of information here and there that in isolation do not mean much, but when you put everything together, you start to see a picture.
A very good clarifying question asked by Teresa Stemeye, who says, SARS-CoV-2, is this something different from COVID-19, or is it the same thing? I think this one's for you.
Yeah, it's interesting. The virus itself is actually called SARS-CoV-2. 2 because we had a SARS-CoV-1 as well, but now this particular virus is the SARS-CoV-2, and it causes the disease COVID-19. So sometimes people change these up, but strictly speaking, the virus is SARS-CoV-2, the disease is COVID-19. But you know, in these kind of discussions, if you say COVID-19 virus, it's fine. Yeah.
We won't dock people points for that.
Exactly.
And it's called SARS-CoV-2 because it's actually close to SARS-CoV-1, right? And it's not the same virus, but it's close enough that people who name viruses suggested that should be the name of the virus.
Yeah, so viruses have families, and this particular SARS-CoV-2 also fits in the family of coronaviruses. And perhaps I'll use this opportunity also to say a little bit about technique in the laboratory that's also relevant to the origins discussion as well. That's a technique that we call sequencing. So sequencing is actually looking at the genetic material of the virus, so the whole recipe. And then you get, so this particular virus has 30,000 letters. So with sequencing, you read all these letters one by one by one by one by one, and you have, until you have all 30,000 of them, so you know what the composition of the virus looks like. Then you can use these different, what we call sequences, in the laboratory to relate different viruses to each other. So when you do that, you can see the different coronaviruses grouping together. You can see other viruses not grouping with them. So this gives you an indication of where this virus belongs. The sequencing is also important if you want to know about these changes, because how do you actually find these changes? You actually need to know what the virus looks like at the molecular level, or with all these letters, to see if it has changed from before. And then you know that this virus has changed or has had what we call a mutation. So it's a really important technique, and we have around like 200,000 sequences submitted already from all over the world, and it has been a really global effort and collaborative undertaking to get this all done, and it's really very impressive.
This is a follow-up question. It comes from LinkedIn. There's been a couple of other similar ones. How many variants or subtypes of SARS-CoV-2 have been identified until now?
Yeah, it's all these variants. I mean, if you really think of it, there are many variants because the virus changes. Some of them survive, others don't. But we have seen that when you do this type of grouping of the virus, even within SARS-CoV-2, there you can see like several different clusters or clades appearing. They're not very different from each other, but you can still distinguish the different groups. We're still discussing how best to delineate these groups, because different technical groups use different terms for that. But generally speaking, we can see what we call the clades appearing. And
there have been sort of, I hate to use the word trends, but there is a sort of a certain variation of COVID that came along earlier and became over the past many months more the dominant one that we see around. Yeah.
So this one actually, people who are actually watching us online may actually have heard about this particular change. It's called the D614G. So what that means is the D that used to be there, that letter I was talking about, at position 614, became a G. So that's the change. This virus, we didn't see it early on in the pandemic, actually very little. But as soon as the pandemic progressed and the virus has spread from China to Europe to US, you see that this virus slowly but surely took over, and it is now the most predominant virus that we can see globally. This has, researchers has been focusing on this particular virus. So early on we looked at, for example, what I just said, these sequences, and we can slowly see this particular virus becoming more dominant. So then the research goes to the next step. So in the lab we look at what does this change mean in the lab? Like, how does it make more copies of itself? How easy is it to get into the cells and those kind of things? And as we progress, we can also see studies in animals, for example, going from one ferret with that virus to another. So we can see that the virus seems to replicate faster in the laboratory, also transmit between animal models. But so far, we haven't seen any differences in clinical presentation of this virus in the humans, or it also seems to not affect vaccine efficacy as well. So
clinical presentation meaning how sick it
makes
people.
Exactly. Yeah, that's right.
It's a virus that can make some people very sick, but it hasn't seemed to affect a different part of the population or be even worse than it already is in a way. Is that what you're saying?
That's right.
Yeah. Peter, some questions on the animal side again. So Mode Mohiuddin is asking, is there any case of an animal dying from COVID-19?
Yes, for example, the mink are dying, getting sick and dying from the disease in a very similar way the humans are doing. Otherwise, very few animals have been shown to be susceptible to the virus or being able to be infected by the virus. So we only have a few examples of cats and dogs and a couple of lions and tigers in the zoo. So we don't really have a lot of animals to study apart from the mink. And in the mink population, as I said, we see animal dying.
But we're looking, right? It's not that a few scientists have looked at tigers and lions and a few have looked at mink, and therefore we know about how it affects those animals. My understanding is that scientists around the world are looking at various animal populations. Can you tell me a bit more about that?
Yes, of course, we are in the search for the animal origin of this virus. We are also looking at different animal species to see if they are susceptible to the virus in the same way we are. So there are studies being conducted on all kinds of animals, and in addition, and of course, focusing on the animals with whom we are more in contact with on a regular basis, and that would be all the farm animals, pigs, cattle, sheep, etc. But also the animals being raised for commercial purposes, such as the mink and the ferrets and foxes and so on. And of course also the other animals we're in contact with as companion animals, our pets. A lot of our pet species are also being studied. So all these animals with whom we are in regular contact are of interest to us because we want to protect them from being infected by the humans, and we also want to make sure that we don't create a risk for us again by creating a new reservoir for the virus in a new animal species.
This is a slightly different question, and it's a bit more of a big picture, but I think it's for you, Frank. This is a question that came a little while back on LinkedIn. If I understand correctly, the 1918-1919 influenza virus, the one that caused that giant pandemic back then, changed to be more dangerous in the second wave. What was the nature of that mutation, if we know? I don't think we're going to be able to revisit that history, but we'll take the second part of the question. What's the likelihood that there would be a similar change in another wave of COVID-19? Do we have a way of estimating percentage chances of change?
The question is an interesting one, and it's important to also compare actually the 1918 virus, which is an influenza virus, to the SARS-CoV-2 virus. Influenza actually behaves slightly differently than COVID-19. Influenza can change in big chunks, so to say. It can change the way it looks drastically. So then when it changes that much and it goes into the human population, most of our immune systems or our bodies are unable to recognize it, and we're still susceptible. So that can cause a huge wave of new disease. As I just explained, the virus that causes COVID-19, SARS-CoV-2, changes much more gradually. And we as scientists call that drift. So the virus slowly drifts away into other different changes. So the chances that SARS-CoV-2 makes a dramatic shift like we had seen in the 1918 virus are unlikely.
Unlikely given current circumstances,
yes.
Unlikely if it's mostly within the human population,
exactly.
All those other provisos as well.
And also the nature of the virus itself.
Because it's a coronavirus, or because of this particular virus?
It behaves differently than an influenza virus.
Yeah. I just wanted to make a point of clarification as well. Something we hear Dr. Mike Ryan say often in press conferences is, because people do ask, has the virus changed? Is that why there's been a second wave? Is that why some countries are seeing a third wave? Is it because changes in the virus? And our best analysis is that no, it's more about human behavior, what we're able to implement, the kind of measures that countries and communities are able to implement, because some countries have been able to bring it under control and it's been much tougher in others. So Dr. Mike Ryan will always say it's about what we're doing. It's not so much about the virus itself. We have a kind of a good sense of what this character is and how to deal with it. A question here, it's quite specific actually, from, hang on here, from Olika Drobnjak-Tomasek. Is there any chance to investigate the pathways of genetic mutation of SARS-CoV-2 and to inhibit that process?
To investigate the pathway of the changes, well, the pathway of the changes, I'm thinking about your question. The pathway of the changes is something that we can follow with the sequences that we get. And then using the genetic material to inhibit. It's a tricky question. I think there's some therapeutics that are coming to the market that target how the virus replicates. There's some other inhibitors that target the DNA sequence of the virus. But yeah, I think we're looking into that. It's still too early to really tell, to give you clear answers for that.
And I think jumping back to something Mike says is the more people it's in, the more chance it has to change. The more it goes back into the animal environment, the more chance it has to change. So the way we control it from changing is to protect ourselves from getting the disease and passing it to others.
That's right.
In a way,
Yeah.
It's philosophical in a way, but
Yeah.
it
It doesn't change that much, but it has a lot of opportunities for selection because it gets transmitted so often to so many millions of people.
And I also remember when we were discussing a couple of weeks ago, you were saying that even within a person, there could be differences in the virus. So it's not that the virus enters you and copies itself identically within you. Is that right?
Yeah, no, that's right. And we see that a lot also with RNA viruses. They're actually quite sloppy viruses. So when they make a copy of themselves, there are a few errors here and there. Most of these errors don't make any impact. They may even be detrimental to the virus. But we call that sort of quasi-species. So there's always a swarm of different viruses, still very similar, but slightly different, so that whenever the virus encounters a change, it can quickly adapt to its new environment.
Peter, back to you and back to what we were talking about earlier as well. We talked about the importance of doing those initial studies. Can you just explain why? So is it just, again, philosophical and historical to understand how this happened, just so that we know, or is there an overarching public health reason that it's important to understand the origin?
There are several reasons why finding the source is very important. The first one is to try to prevent the next one from happening. Nobody wants to go through another pandemic like this one in the coming years. Unfortunately, they happen on a regular basis, and looking back in history, we have seen pandemics on a regular basis. But we have never been able to get a good understanding of how they emerge and how they happen and where, and we need to try to understand that to help us prevent the next one, but also to help us prevent this one for coming back at us once we have dealt with the current outbreak in the human population. We don't want to keep an unknown human reservoir somewhere out there,
Or
and
animal
reservoir where the virus will just stay dormant and then come back at us, perhaps in a different versions. So these are the key two reasons. The third important reason is that if we can find the version of the virus before it adapted fully to humans, it could give us some very interesting clues about how we should better design vaccine, therapeutics, and so on, because we will have a better understanding of what type of changes in the virus are important for human adaptation, for human infection, etc. So these are the key reasons why it's so important to find the source.
Is this what, I've heard members of the team talk about, most recent common ancestor? You're basically trying to find the Adam and Eve, or the Adam or Eve for this virus in a way?
Yes, we try to find how it looked like before it adapted to humans.
Before it started to change?
Yeah.
And when you talk about an animal reservoir, you mean you want to find, so studying the virus, you said that it looks like it came somehow from a bat, then passed through another animal likely before it came to humans. Is it that other animal that we're trying to identify?
Yeah, there are two possible key scenarios. One is that it jumped directly from a bat to a human and was either fully adapted or adapted very quickly to the human population. While possible, it's an still unlikely scenario. The other more likely scenario is that it's jumped from bat to one or several animal species, where it had time and opportunities to adapt to these new environments, and from there again jumped to human and adapted to humans.
So WHO has been mandated by our member states, the emergency committee that recommended that this outbreak be declared a public health emergency. They've all asked WHO to lead in these studies to understand the beginning and how this came to be. Can you tell us how those studies are going? Where are we in the evolution of these studies to understand the history and what the next steps would be?
Different set of studies in parallel. One is to understand and look at all the studies that have already been done since the beginning of this event in the direction of understanding the source. Second one is to start additional studies, the one that we have developed in the research plan for the origin studies, and these will start in and around Wuhan, and that's the one I described already, looking at what happened in the markets in December 19, what happened among the first initial cases, what information we can get out of these studies. And the third set of studies is to, and these studies are first now being undertaken, and then we also have a series of studies that have been ongoing for a while now to understand what animal species are susceptible to the virus, which one are not, how easily the virus transmits in certain animal species compared to others, etc., because that will also help direct future studies on where we should look for the origin. Should we look in a farmed animal? Should we look in a wild animal? Should we look in a certain type of mammals or in another type of mammals? So all these studies give us each some clue and to better guide us into where the source might be out there.
Some people will say we're 11 months into this pandemic. Why don't we have that answer already? How does this follow-up work and study and research compare to MERS and SARS, which are other coronaviruses? Are we on a similar timeline? Are we faster? Are we slower?
These are complicated studies, and they take time. That's one issue. The second issue is that remember how Wuhan looked like back in January, February, and March this year. The city was battling with severe outbreaks. The city was in lockdown for a very long period of time. All the people available and the logistic available in the city was geared towards combating the outbreak there. So there was no opportunity, no resources to do these more long-term studies into the source of the. So we had to wait until the situation was more calm and in a way in Wuhan. With regards to other similar events, yes, it's a long time since this started. Ideally, we should have done many of these studies immediately when the event started in December, January last year. But with SARS-1 back in 2003, it took months, and it took more than a few years before we had a good understanding of the intermediate animal host that was the source of SARS, and of the link to the original source in bat population. That took many years before these links were made, and we never found the virus in any of these animal species. It was just a strong suspicious that we could reach. With MERS that started in 2012 in the Middle East, it took more than a year before we made the link with the exposure to camels, and camels being the source of this virus. And that was a little bit by chance. I remember we were called for by colleagues in Qatar who told us, look, we have this farm where we have 2 cases that are sick with MERS. We think that the source might be on that farm. Can you come and work with us on that? So we were immediately on the ground, and by sampling and searching that farms and all the animals on that farms, we managed to make the first link to camels and got that confirmed later on. So it took about a year to get that first indication. So this time we're not even a year through this massive pandemic. So it's late, but it's not too late. I'm confident that we still have very good opportunities to learn much more on the origin of the virus.
Just two quick questions for you, for your area of expertise. Nick Aina Nadhira is asking, is there a possibility that livestock or pets might actually need to be vaccinated sometime in the future?
That's a good question, and probably short answer would be yes, because we know that cats and to some extent dogs are susceptible to the virus. So if the virus is still circulating in the future around us, it would be a good idea to also be able to protect them in the same way we're trying to protect humans by developing vaccines for humans. And for farmed animals, for domestic animals, the same. We know that some species may be slightly susceptible. They're not as susceptible as what we see in the mink, so it's totally different scenario, but they could become more susceptible again if the virus change. So it might be also a good idea to be able to vaccinate pigs and other animals in the future. They do not get sick, they do not transmit the disease easily, but in some cases they may be able to get infected by the virus. And again, we don't want that to happen too often with the animals we're in contact with.
So while all these experts are working away on developing a human vaccine, would you estimate that in parallel there are animal health specialists who would already be working on a vaccine for animals, or would they have to wait for a human vaccine to be ready and would somehow work around that one to make an animal one?
The two efforts could run in parallel, but once we will have a vaccine for humans, it will be much easier to adapt to even use the same vaccine for animals. So there is no reason to duplicate efforts, in particular in the first phase of the development of a vaccine.
And another question for you, this is from Periscope, from Jane Gabbard, who wants to know, would stopping human encroachment on wildlife environments help prevent this kind of outbreak? So it's really a looking towards the future kind of question.
It's true that many of these new emerging diseases that we get from contact with animals, in particular with wild animals, are of concern, and therefore the logical quick fix in a way would be, well, let's stop to get in contact with wild animals. The problem is that that's a very difficult thing to achieve. But it's true that the more we move into areas and establish ourselves and establish settlements in wild environments, the more we get in contact with new viruses and bacteria and the like. And therefore, we increase the risk of these disease jumping to the human. So managing better our interaction with our environment, in particular with pristine nature areas where we have not previously been actively establishing ourselves, is probably a good idea. Unfortunately, we are growing in number on the planet. We are taking over bigger and bigger parts of the landscape around us, and therefore it will be always a balance between what we should and what we can, and will be also depending on how good we are at managing our environments and our interaction with the environment. Very complicated topics.
Yeah, it's really about how humans, how we care for this planet in a way. I'm going to wrap up now. Thank you both, Frank Konings and Peter Benenbarek, for being with us today. If you have follow-up questions, you can drop those in the comments too. That helps us guide what kind of products we need to develop for you. And now I'm going to round up the names of some of the countries where you've watched us from today. Sri Lanka, Denmark, Kenya, South Africa, Argentina, UAE, Cambodia, Pakistan, Bhutan, Jordan, USA, Sudan, Bangladesh, Nepal, Nigeria, Tanzania, Canada, a few other countries from LinkedIn: Greece, Rwanda, Mauritania, Saudi Arabia, Cameroon, Morocco, Uzbekistan, Liberia, Spain, Poland, and many others. Thank you so much for tuning in today. We really appreciate your interest in the work that we're doing. Please keep tuning in. We will do these regularly. Let us know if there's a topic we haven't covered and that you'd like to hear more about. Rounding it out, my name is Nika Alexander from WHO headquarters here in Geneva.